So pharmaceutical companies need to figure out how the drug is going to get to the bacteria. So, but for our sense and purposes, it's just an outer protective layer. We'll just not stress about it. It's not ever drawn. Nobody ever talks about it other than just learning the anatomy. So we'll just forget about it. So we can look at most bacteria and all bacteria, but let's just draw a couple of bacteria. So we'll draw a rod. This is a bacillus. And we'll draw a cocci. We're gonna say this one is gram positive and this one is gram negative, but don't forget that a gram, they can be either or. So there are gram negatives that are cocci, and there are gram positives that are rods. We'll see some of those in the microscope, but this is just the most common. But there are actually a lot of gram positives that are rods. There are a few gram negatives that are cocci. So just so you know, this is kind of traditionally how we draw them, but don't forget it can flip-flop. This is not exclusive. So gram positives, what color are gram positives?
Purple.
Purple, nice. What color are gram negatives?
Red or pink.
Red, good, yes. So yeah, they're pink, but I just like to say red just because it starts with a P. You don't like to say it starts with a P. But if you think of like negative red, it's just kind of a blurry red. Okay, so starting with the innermost layer, I really wish I had colors, but starting with the innermost layer here is the cytoplasmic membrane. And the cytoplasmic membrane is made up of phospholipids. PPL stands for phospholipids. Oh, thank you so much.
Yeah, I just got one of each.
Oh, perfect. Thank you, thank you. Is phospholipids. So that phospholipids, remember, they look like this. And all of our cells and membranes, all in organisms the world over, have this arrangement outside of their cells. And the reason is because it's the phosphate plus the lipid automatically forms a bubble. It just forms a bubble. And our cells have used that, the chemistry of that, to make the bubble around, to make a little protective layer around our organelles and around our cells. So if you zoomed in to like one of our cells, for example, I always try and draw our cells like this. This is like a little epithelial cell. Sometimes cells are drawn like that. You know, traditional cells are drawn like a circle. But our own cells, I like to draw them like this so you know what I'm talking about. This is an epithelial cell. It's a columnar cell. You have a nucleus, you have an endoplasmic reticulum, you have a Golgi, you have ribosomes, all that kind of stuff. So if you zoomed in on this little membrane right here, it's that. It's a phospholipid bilayer. And that phospholipid bilayer is called semi-permeable. That means it's permeable only a little bit. So some stuff can come in and some stuff can come out. It's selective about which stuff it lets in and out. Cells are very protective. They don't want random stuff inside the cell, right? We have to protect ourselves from stuff getting inside. So we have this semi-permeable membrane where water can come in and out, non-polar molecules can come in and out, but we're very protective about what comes in and out of our cells. So same with a cytoplasmic membrane of bacteria. It's a selective semi-permeable membrane. They're protective. It's a protective layer. Lets stuff in, stuff out. Some stuff in, some stuff out. So that's the cytoplasmic membrane.
Yeah, I'll check it out.
Okay. If we're talking about the cytoplasmic membrane, then what if this is a gram positive and this whole thing right here, what is this layer called? This thick layer.
That's the word that I can't say. It starts with E.
Yeah, What is the name of the structure? that's a cell wall. Very good.
Peptidoglycan.
Yeah, nice. There you go. Peptidoglycan. Good. Peptidoglycan. So we know this is not an archaea. We know this is not a human cell. We know this is nothing else because only bacteria have peptidoglycan, and all bacteria have peptidoglycan. Almost all. So I'm gonna just erase this color because I'm gonna be consistent with my color.
So the gram-negative has the 3 layers, right?
Yep.
Gram-positive has the 2 layers.
Yep.
With the cytoplasmic membrane inside and the cell wall on the outside. And
Yep.
the gram-negative has the cytoplasmic membrane, the cell wall, and then the cell membrane.
And then the outer membrane. Yep. Perfect. So this is a sandwich. The gram-negatives are a little sandwich. Yeah.
What kind of a category is a peptidoglycan molecule? Is it a sugar?
Peptidoglycans, so glycan tells you it is a sugar, and peptido is a protein. They're protein sugars.
Okay.
Yeah. And so those are the NAGs and NAMs it has on the slide, like glucosaminoglycans. You can look at the structure if it helps you just to
Yeah,
remember it. Yeah.
it would.
Yeah, awesome. Okay, so here is the outer membrane. And then, okay, so here, those are our layers. That's it. That's it. Basically. I'm gonna go over one more layer later on. Don't forget there's inside, there's the nucleoid, is the DNA. There's an area of DNA that's called a nucleoid. There are plasmids, these circular DNA pieces. There are ribosomes that make proteins. Plasmids, we haven't really talked about what plasmids do yet. We'll talk about that later on in the semester. Let me see what else I want to address before we move outside. So here's a nucleoid region. Here are some plasmids. It has some ribosomes. It has things that are called vacuoles. So vacuoles are like little, sometimes they're called lysosomes depending on what their function is, they have a different name. They have like peroxisomes, lysosomes, vacuoles, inclusion bodies. They have different little terms depending on what they're holding, what's on the inside. Just for general purposes, we can call them all vacuoles. That's fine. Yeah.
How do you want us to differentiate between the 2 circles?
So you will hopefully be able to see that this is just a ring of DNA. So hopefully, like if you ever have a question about that, ask. But vacuoles are spheres.
Yeah.
And then plasmids, even though I'm drawing them like this, they usually kind of look like a little mini this.
Okay.
Yeah, so they're like a little piece of DNA that's just
They're like
stuck in there.
SpaghettiOs?
Yes, like
Okay.
the little SpaghettiOs of the main spaghetti mess. I love that.
Thank you.
Yeah, because I talk about this spaghetti mess a lot because we're going to talk about how it unravels and all that kind of stuff. So, okay, so those are plasmids. Those are ribosomes. What else am I missing? Ribosomes, cytosol, S-layer, plasmid. Okay, so let's talk about what's on the outside. So don't forget that this outer membrane of gram-negative bacteria is a phospholipid bilayer. This is a PPL. This does have a phospholipid bilayer, but attached to those phospholipids are lipopolysaccharides. So there's like these little tiny things called LPS, lipopolysaccharides. I should probably write that out so that everybody knows. Lipopolysaccharides. So poly means many, saccharide means sugar, lipo means lipid. I don't care that you know that, but just know that it's a lipopolysaccharide. And you will absolutely need to know this for all quizzes and exams because this is why gram negatives are so toxic. So gram-negatives, they have this outer membrane and they have these lipopolysaccharides in their outer membrane. And what will happen is our immune system, our body, our white blood cells will come and chew these up and spit them out and chew them up into the pieces like our white blood cells do. Our white blood cells are really good at chewing stuff up and spitting things out. So when it chews it up and spits it out, now there's lipopolysaccharides in our bloodstream and lipopolysaccharides everywhere, and they're still toxic even when they're not attached. So that's why we worry about gram-negatives also, and sometimes why when you're treated with antibiotics or a patient is treated with antibiotics, they still don't feel good. And it takes them a while for their body to get rid of those lipopolysaccharides because the antibiotics are killing the bacteria, but those lipopolysaccharides are just hanging out, still making your patient sick. So, okay, so that's LPS. So don't get confused with what are called fimbriae. So fimbriae are a little longer. So these are the hairs. So fimbriae are the little hairs, and these are for adherence. They stick to stuff. And so those are, let me go to my most. Not every bacteria has fimbriae. So not every bacteria has fimbriae, but a lot of them do. So they're adhesion or sticky. And then some have what's called a pilus. So let me draw the flagella longer so you don't get confused between a flagella and flagellum. So flagella, some bacteria can have a bunch of these. Some bacteria can have them on both sides. Some can have one, some can have both, some can have many. The flagella, those are for motility or movement. Spell that right? Okay. Sorry, did you have a question? No, you're good? Okay. Okay, so flagella are long and those are for motility. But hopefully it will be apparent, and I'll make sure that I pick a good picture, that there's something called a sex pilus. And that is for what we call conjugation. And it seems weird that bacteria have sex. They don't really, but they do, they can pass DNA to other bacteria, which, I mean, I guess is a method of sex, is kind of the point. But I'll tell you what that means when we get to that section. We do a whole section on bacterial reproduction. Okay. So that is it for bacterial structure that we've gone over so far. Does anybody have any more questions about that? No? Okay, so we're going to go over what a glycocalyx is. We're going to go over what an endospore is. There are really good pictures online. This is really cool too, is different bacterial shapes and different types of bacteria. It's just a cool website if you want to check it out, just if it helps you to kind of watch some stuff. Okay, yeah, so we'll continue there. Is there anything else we need to talk about? Oh, so yeah, just don't forget about the gram-negatives. They're the proteobacteria and Bacteroidetes. The actinobacteria and the firmicutes are the gram-negatives, or sorry, the gram-positives. Yeah.
Will you be marking down if you get spelling wrong on them?
No, I don't care about spelling. As long as I know it's like peptido something, as long as it looks kind of like the word, you get full points.
I have a quick question.
Yeah.
Do only gram-negatives have the fibria sticks coming off of the outer membrane, or do gram-positives too?
Yeah, gram-positives will too. So here on the slides, if it helps you, I put, so outer membrane is gram-negative only. Then the fimbriae can be gram-positive or gram-negative, but only some species have those. The pilus is mostly gram-negative for the most part. But I don't wanna say that I'm not gonna ask you that, but I really, it just know, as long as you know that some have it and some don't. But the characteristics that you do need to know that are specific to gram-negative are the outer membrane lipopolysaccharides, because that's what qualifies the gram-negative as gram-negative. Does that make sense?
Yeah.
Okay, awesome, yeah.
And the importance of the gram negative and gram positive is so you're knowing like how to fight it basically.
Yeah, very good point. So I will talk about, later, like, in a couple of lectures, we're gonna talk about, cytopathology, like what the bacteria is doing to harm our cells and how it harms our cells. So peptidoglycan itself can be harmful also. Lipopolysaccharides are definitely harmful, but they spit out things like toxins. So I'll talk about the toxins that they spit out. And then, also we'll talk about antibiotics and the different targets of different antibiotics. So targets for antibiotics, I'll kind of tell you, like, give you the end of the story already at the beginning of the story, because it's kind of cool to figure it out as you go along with antibiotics. But if you look at like their ribosomes, you know, you have to have a target that you're not targeting human cells as well. So when you're taking a pharmaceutical, you don't want it to be harming human cells as well. So you have to like look for those qualifications that make the bacteria unique, so that the pharmaceutical doesn't harm our cells as well. Does that make sense? Okay, cool. Yeah, so targets for sure, but also how they can make us sick. I'll show you that. Cool. So do you guys have a little bit better of a picture of bacteria? Kind of what, there's two main types, gram-positive, gram-negative, but there's all kinds of different shapes. So don't forget, these gram-positives and gram-negative can come in the spirochetes, in the spirulia, in the vibrio, in the, streptococci, staphylococci, diplococci, streptobacilli, diplobacilli. They can come in the different arrangements and the different shapes, but they are generally, you put them in two different categories based on their thickness of their peptidoglycan layer and the outer membrane. Good? Anybody else have any more questions? So what I'm gonna do, actually, do you still have the keys, Dr. Rosnier, the key to the exam? Oh, I'm sorry, Mom, I should mention that.
Yeah.